Hello, hi guys. Am I audible? Am I audible? Uh, Hello, am I audible? And is my screen visible?
Yes,
Okay. Uh, let's wait for 1 or 2 minutes for more people to join and then we'll begin with today's session. Okay.
yes, I'm sure.
Okay, so let's begin today's session. I am Gitika Agarwal. I'm a PhD scholar at Center for Ecological Sciences in ISC Bangalore. I will be taking this tutorial in place of Shakur. He mostly takes the tutorials scheduled on Mondays. So, we are going to be discussing the fifth week of Wildlife Ecology course of NPTEL. Let's keep it an interactive session. There are just a few of us here, so you can please stop me anytime you have a doubt or write your query in the chat box. And if you have anything you want to discuss from the quizzes, please let me know and we'll talk about it. Okay, so in this week of Wildlife Ecology, we were discussing about population ecology. So let me ask you this: what is a population? You all would know right by now because we have like talked plenty about it. So what is a population? You can write it in the chat box or you can unmute yourself and tell me. So what is a population and why do you think we should study populations?
Population means the amount of number of species, like what to say, like humans, we have a number of we are a number of group of species like monkeys. I mean apes. They are group of species. So the amount, how many of them they are? This means populations.
Okay, Mohammed, you're partly correct. So basically, population is a group of organisms that are living in a same geographical area and they belong to the same species. So that means that they have the capability to interbreed. So it is the number of organisms, but all of this group of organisms should belong to the same species and they should live in the same geographical area. They should share the habitat and be able to interpret. And why do you think it's important to study these populations? Like, how can that help us? Anybody who would like to venture? So, okay, basically, we study a population for many reasons, but some of them would be to understand what are the natural phenomena that are going around in that species. For example, what is the breeding ecology of the species? How do they forage? What do they forage on? Where do they live? All these things you can understand by looking at different populations. Then it could be to regulate and manage different species. So if certain species is endangered or certain species is important to a nation, for example, tigers are important to us as they're a national animal. So you would want to regulate their populations, you would want to manage populations at a good number of organisms. And it is also to know how different factors, may they be extrinsic factors or intrinsic factors, how they are affecting the individuals of the population. Okay, extrinsic factors, for example, the temperature, for example, the landscape, the humidity, the amount of rainfall, precipitation in that area. And intrinsic factors like what are they feeding on, what is their breeding ecology, all those things are the intrinsic factors. And their behaviors amongst each other or with the environment, how are these different factors affecting the population. So why do we need to carry out the assessment of a population, or why do we need to know what are the numbers of population in a given area? Any guesses?
Like to know the interaction between different population species.
Yes, to know the interaction of these population with other populations. But why do you need to know the number? Like how many individuals are there in this population of a particular species? So basically, you would want to know that because you would want to know whether the population size, if it's increasing or the population size is declining over time or is it stable, the population size in that particular area. Then, if you are comparing different areas, you can see that okay, in area A, the population size is increasing, but in B, it is decreasing. That you can find out what are the differences in these two areas or in these two regions, and what are the reasons that are actually causing this increase or decline in the number of individuals in the population in these two areas. Or you would want to make a decision, how do you want to intervene with this particular population. So if it's an invasive species and the population number is increasing, then you would want to intervene in a way so that you can reduce this population so that it's not harming the native species. There you would want to decline or you would want to decrease the number of individuals in that population. But if it's an endangered species, if it's an endemic species, you would want to increase their numbers. So it helps you to make a decision of how you would want to go about managing the population. And you would also want to determine what are the various threats to this population. As I mentioned, in some areas the population might be flourishing or thriving, and in other areas it might be declining. Why is that? Is it because of increased hunting in some areas? Is it because of the low resources? Is it because of the temperature conditions? Is it because their habitat is being degraded? Some of these reasons. So you would like to know the factors that are affecting your population. Or you would also like to check what is the efficacy of the interventions that you've applied to a particular population. So say there's a population of tigers and you're trying to increase the number of tiger individuals in that particular area. So what do? You think that okay, fine, we'll introduce more prey that will help the tigers get more resources and increase the population. But so you increase the prey there, you increase the amount of deer in that area. So after a certain amount of time, you would also want to see what is the efficiency of these interventions. Did the population actually increase, or is it going down, or what is happening, or is it still unaffected by the increase in the amount of deers? So these are some of the reasons that you would want, that you for which you carry out the assessment of a population. And India carries out a lot of population surveys. Some of the famous ones would be the status of the tigers in India. So a recent report was given out in 2022 where they are mentioning what is the current status or how many tigers are there in the country. Since it is our national animal, everybody is interested and wants to increase the amount of tigers in the country. State of India's birds is another organization or is another team that carries out the surveys of Indian birds, and they try to figure out which birds are thriving, which birds are stable, and which birds are critically endangered or endangered in India, and where they are found, what are the habitats, what is their ecology, all these things. And we also see on the left here that there are some things like the leopard count happening here in different states. So a lot of different organisms population surveys are carried out from time to time, so as to figure out which populations are going up, which are going down, and what are the factors behind these. So what do you think are the factors that affect population sizes in a given area? I've already mentioned it in the previous slide, so can you please tell me what are the factors that affect the size of a particular population?
Amount of food,
Hmm. Amount of resources, yes.
climate,
Climate,
the
temperature,
environment.
yes. And what? There would be a predation pressure. What is the predation pressure in that area? What is the habitat? Is it if the habitat is a forest, but humans have been, deteriorating the forests or cutting them down, then all these factors will affect population sizes. Some things, so demographic stochasticity is something that is a characteristic of the population. So what is the birth rate in the population, what is the death rate, what is the sex ratio, how many males, how many females, because if there are a few females, then that will not lead to an increase in the population, because females are the ones that are going to give rise to the progeny. Then there is going to be environmental stochasticity. There could be a sudden catastrophic events like droughts, floods, spread of disease, these things. There could be genetic problems in the population. For example, there could be a founder effect leading to inbreeding depression. Only a few organisms that are mating within the population, leading to an inbreeding depression. There could be genetic drift. There could be a loss of heterozygosity in the genes in that population. And there could be certain behavioral issues. For example, what was mentioned in the lecture was, say there is a pack hunting group of wolves. Okay, they hunt in a pack. So if the population size goes below a certain number, then there will not be enough individuals to go out on a hunt, and that is going to reduce their hunting efficiency, and in turn it is going to reduce the resources that they can gather to keep on surviving, to keep on sustaining their own population. So these are some factors that affect the population sizes. And there are certain population parameters that we want to calculate when we go on assessing a population. So, can you tell me what are the basic parameters that you would like to measure when you go out and you assess the population? Or how would you describe a population?
By sex ratio, ma'am.
Yeah, there is the sex ratio, but before knowing the sex ratio, there's something even more basic than that you would like to know before you can reach the sex ratio. Anybody? So, okay, so very basic parameters of population when you go out and look at a group of organisms would be the size of the population, what is the number of individuals in that area, and as you mentioned, the sex ratio would come from this. When you measure the number of individuals, you would measure the number of males and the number of females, and then you can find out what is the sex ratio. And the second important thing is the population density. What is the number of individuals in the area, in the population per unit area? And so this is the basic difference between the population size and the density. Size is the number of individuals in the population. Density is the number of individuals per unit area. And there are two basic types of population density. One is an absolute density and one is a relative density. So can anybody tell me what is the difference between the absolute population density and the relative population density? Anyone? You can type it out in the chat box, or you can just tell me like this. What is the difference between the absolute density and the relative density of populations? No? So absolute density would be when you go out to a particular geographical area and you either totally count the number of individuals in that population, or you sample that population and you reach a certain and you measure the population size by this indirect sampling, and then you calculate the density of that particular population. And relative population density is when you are comparing two different populations and you just want to say that, okay, fine, this population is more dense as compared to population B. Population A is more dense as compared to population B. This you can do just by looking at the how frequently uh, are you coming across the individuals in a population when you go into that particular area. So in the absolute density, you absolutely measure the population density of that particular area by total counting or by sampling, and in the relative density, you just compare the densities of two different populations. So, some other population parameters that we measure in a population are age pyramids, crude birth rate and death rate, general fertility rate, age specific fertility, total fertility rate, replacement level fertility, juvenile mortality, life expectancy, physiological longevity, ecological longevity, immigration, emigration, and the population growth rate. So, can anybody tell me what is life expectancy? That should be very basic.
The average life of an of a
Yes,
animal,
exactly.
like
So,
how
what is
much
the
is expected
average
to live.
lifespan of individuals in that population that you would refer to as the life expectancy? And what is the physiological longevity versus the ecological longevity? What is the difference between these two? Anyone? Physiological versus ecological longevity.
Physiological has something to do with the biology itself, biology of the animal itself. But
Yes,
the ecology has something to do with the environment or the habitat of which it lives in.
exactly. You're correct. So, physiological longevity is something to do with the biology, exactly. So, how long can that organism survive under ideal conditions? And ecological longevity is how long does an animal actually survive in the ecological setting in which it is found. So, ecological longevity would in a sense be the realized physiological longevity. Physiological longevity is the lifespan under ideal conditions, and ecological is in the given ecological setting where you have predators, you have limited food resources, all those things happening. Okay, so moving on, let's discuss how do we measure the different population sizes. So, three basic ways of how to measure population sizes are total counts or a census. Or you can carry out sampling. You can either do a quadrat sampling, or you can do a capture recapture, or you can do a removal. So, can you tell me what is the total count or what is the census? Like that is very basic. Everybody would know what is a total count.
The total count of an organism over a geographical area, including all types of genders.
Okay, so what? How do you carry out the total count? Okay, so by total count, we basically mean that yes, you go out in the field or in that region, and you are counting each and every individual that belongs to that particular population. So you go, like the human censuses happen in a country. So they come to each and every household and they count every single person that is present in that house. So similarly, for a population census, you would go out in that area in which the population is found, and you would count each and every individual belonging to that population. So, do you think it is a feasible method, or is it uh, Is it possible to do total counts every time for all populations?
Not really.
Yeah, so total counts are not really that feasible. And so what we do instead is we carry out sampling. Sampling is basically you take out a sample from the population, and you do it in such a way that the sample is representative of the population. So you can either carry out quadrat samplings, in which you divide the region in which the population is found into different quadrats, and you select certain specific quadrats that you will visit, and there you will count the number of individuals in the population, and from that you will extrapolate what will be the population size in the entire area. For example, if you divide your region in 10 quadrats and you just visit half of them, you visit 5 quadrats, and you found 2 individuals each in each quadrat. So you found 10 individuals. So you would say that the other quadrats will also have on an average 2 individuals, and you will say that the entire population would be of about 20 individuals in that region. And so, okay, so tell me, what is a capture recapture method? What do you understand when we talk about the capture recapture method of population survey? You would know if you watched the lecture videos of this week. What is the capture recapture method of population survey? Nobody? So, okay, in capture recapture, what basically happens is you go out, you capture a few individuals from that population, you mark them, and then you release them back into the population. And you let some time pass so that these marked individuals, they get mixed with the other individuals of the populations. And then after a few days, you go out and you capture again, or you set up your trap again. And in that trap, how many marked individuals you get as compared to the total amount of individuals in the population. That would give you an estimate of what is the total population of that area. I will go into the details of this a few slides later. And the removal method basically tells you how many organisms you. This is an invasive method. It is not very frequently carried out, and what you do in this is you go out, you remove a certain number of individuals from the population, and the number of individuals that you can remove from the population each time you go would be proportional to the amount of individuals that are present in the total population. And moving on, so when you're carrying out sampling, because total counts are not feasible and removal should not be practiced much because it's an invasive species, so what we're left with is sampling. So can you tell me what are sampling units? How do you define a sampling unit? Anyone? Nobody? So a sampling unit would be the identifiable element of that sampling area that you want to uh, go out and measure the population size from. So, for example, in this quadrat method, each quadrat is a sampling unit. So, each quadrat is an identifiable piece of land, or an identifiable area, and you can sample a few of these quadrats from the entire region. So, that is called a sampling unit. So the entire area could be the forest, could be the grassland, could be the river, and the units that you are going to sample would be called the sampling units. And what is a sampling frame? Does anybody know what is a sampling frame? So a sampling frame is just the list of these sampling units. So, for example, here you go to these quadrats and you choose this quadrat, this quadrat, this quadrat, this quadrat to these areas to sample from. You randomly selected these, and the list of these quadrats 1, 2, 3, and 4, this would be your sampling frame. And so what is the sampling intensity? Anybody, can you define sampling intensity? It is also called sampling intensity or sampling effort. No one? Sampling intensity is basically the ratio of the number of units that you have included in your sample to the number of units that are present in the population. For example, here it's the amount of quadrats that you include in your sample versus the total number of quadrats that are there in your region. So, how many quadrats are there in this figure? So, there are 1, 2, 3, 4, 1, 2, 3, 4, 16 of these, and um, 16 of outside, so 32 in total. And if you choose just these 1, 2, 3, 4, 5, 6 of these, then your sampling intensity would be 6 by 32. And the sampling intensity, if it is 1, that is, then it becomes a total count or a census, because you have sampled all of the area properly for the population size. Okay, so moving on, how do you select your sampling units? How do you decide which of the quadrats in that area are you going to select for sampling? Can anybody tell me, or can anybody guess? Anyone? How do you select a sampling unit? Just take a guess. Nobody? Okay, so it is very simple. Basically, you can either simply randomly sample your quadrats. So from the total areas, from the total rectangles that are present in your region, you just randomly pick up some. This can either be done by a random number generator or just by randomly picking up some of the quadrats out of the total number. Then there is stratified random sampling. So you first divide that into different regions, or say the population is dense in half of the region and less dense in the other half. So you would take randomly sample from within these two halves, but you would not randomly sample from the entire area. So you divide that entire region into some strata, and then you randomly sample from each of the strata. Then there is systematic sampling. Systematic sampling is where you just choose a random number. Say I am starting from the second quadrat, and I will choose every third quadrat starting from the second quadrat. So you'll choose quadrat number 2, number 5, number 8, and so on. Then there is multi-stage sampling. Multi-stage sampling is like you're going to sample within India. From India, you're going to sample certain states, say Gujarat, Madhya Pradesh, and then Karnataka, a few of these states. That is multi-stage. From India, you chose 3 or 4 states. From these states, you're going to choose certain specific regions. So that is going to be your multi-stage sampling. And then the last one is the PPS, probability proportional to size sampling. So in an area where the area is bigger, you're going to choose more samples from that area, and if the area is smaller, you're going to choose less number of samples from that area, so that they have an equal representation in your final sample. So simple random sampling, as you can see here, you just randomly pick up some individuals from the population. Cluster sampling, you take these, for example, they're taking these two clusters. They're not randomly picking certain individuals, they're taking certain clusters from that entire area. Then there is stratified sampling. You divide the area into certain layers, and then you choose randomly from within these strata. Then there is multi-stage sampling. So here they have drawn these people. but you think of these as quadrats. So if these are different quadrats, you're randomly choosing the quadrats. You are choosing a cluster of quadrats. You are dividing them into two different layers and then choosing from that. This is multi-stage. The states within the country, then different regions within that state, and within those regions you choose certain quadrats. And this is the systematic sampling. So you sample every third quadrat from the total number. So these are the various ways of how you choose sampling units from your area of interest. And then moving on to capture, mark, recapture sampling method. This is very popularly used for organisms like there are fish, there are birds, and even the bigger mammals, the terrestrial mammals. You perform this method of capture, mark, and recapture. In this, the different stages include number one, you capture some organisms. Maybe you've set up a trap, maybe there are camera traps, or there are just these different traps for fishes. You perform uh, fishing, or for birds, you set up mist nets, and you capture certain number of organisms from that population. You mark these captured organisms and then you release them back, and you allow them to get mixed with the entire population. And after some time, you again carry out this capturing exercise and you check for the number of individuals that are marked. Then you use this formula, which is the Lincoln Peterson index, in which the population size estimate would be the number of marked individuals that were released, the total number of individuals that you marked basically, multiplied by the size of a second sample, divided by the marked animals or the number of marked animals that were recaptured. So, to understand it more basically, it is just that the number of individuals you marked in the first sample divided by the size of the whole population will be equal to the number of marked individuals in the second sample divided by the total of number of individuals that were caught in the second sample. Is this clear, or do you want me to go over this again? I can explain it in further detail so that this is clear. How do you carry out a capture, mark, recapture? Is it clear to everybody? Should we move on? Okay, I will move on. So, capture, mark, recapture is very regularly practiced in birds. You go out, you set up a mist net, and you get certain birds, you capture some of them, and then you carry out banding, color banding for these captured birds. You can see here there are these colored rings that are put in the tarsus of these birds or the legs of the birds, and you can identify different individuals of the population from these color bands. So you put them in a way, for example, there is white and blue, then in the next one you would put, say, red and black. So you use different combinations of these colored bands which are specific or unique to the different individuals of the population. So these are not all from the same population, these are different species of birds, but I'm just showing how the color banding is done. So you capture the individual, you mark them by color banding, you release them into the population to allow them to mix with the rest of them, and then you recapture. And when you recapture, so you will find certain number of them are marked and certain are unmarked, and you use the formula that I showed on the previous page, and you measure the entire population. You estimate the population size of the entire population. So here is a question for all of you to solve. So there's a scientist who wants to estimate the population of an endangered bird. So he goes out, he places a mist net, and six birds of that population get captured. He marks them using these color banding and releases them. 5 days later, he comes back and he places the mist net in the same area. This time, he captures 24 birds from that population, out of which three are marked. From three of them are marked. That is, three of them were also captured in the previous mist netting session. So you have to estimate the total number of birds in this population. So can you guys tell me how will you do it? So six birds were captured and marked in the first attempt. Then in the next attempt, he captured 24 birds, 3 of which were marked. So if you apply this formula here, so the number of birds that were marked in the first sample is 6, and the number of birds that are marked in the second sample is 3, and the number of birds caught in the second sample is 24. So what will be your answer for the size of the whole population? Guys, can you calculate and tell me what will be the size of the whole population? This is the formula and this was the question. 12. That is not the correct answer. Anybody else? So you capture 6 birds in your first sample and you mark all of them. Second time you capture 24 birds in your sample, out of which 3 of them are marked. So if you apply the formula here, the population size would be number of marked birds, which will be 6, multiplied by size of the second sample 24, divided by 3, number of marked animals that were recaptured. So what would be a population size? 6 into 24 by 3, and if you cut it, it will be 2 multiplied by 24, 48 should be your population size. Did all of you get it? Did all of you get the answer? Okay, so let's move on. So this was capture, mark, recapture, which is very commonly done for birds and fishes, and for other, even for other terrestrial mammals. Now moving on, what are the basic assumptions in mark capture, mark and recapture? What are the basic assumptions uh, that are behind this method of sampling? Does anybody know? Anyone? What are the basic assumptions of mark recapture method? Nobody? Okay, so some of the basic assumptions of mark recapture are that when you perform this mark and recapture, between your two capturing events, there was no immigration or emigration from that population. Because if after your first, say, capture and marking of individuals, you release them back, and some of them emigrate from that area, some of the marked individuals emigrate, then your population estimate will be a little faulty. So there is no immigration or emigration. Then the marked and the unmarked animals, the probability of the marked or the unmarked animals being captured is equal. So marked and the unmarked animals, they get captured randomly. There is no higher probability of either one of them being captured in our trap. Then the mortality rate is also the same for the marked and the unmarked animals. That means that the mark that you have placed on those organisms does not influence them in a way that they die faster or they survive longer or anything like that. So the mortality rate is the equal for both of these organisms. And then these marks should not be lost over time, and they should be easy for the experimenter to spot. You should be able to spot these marks on those organisms. And lastly, the marked and the unmarked individuals should intermingle equally. That means that they should, mix with each other equally in the population. It should not be that the marked individuals get separated out from the other ones and they cannot uh, intersperse between the other ones. So that would also uh, bias your estimate of the mark of the population size. So these are the basic assumptions behind mark recapture. They should be both of these types of organisms, marked as well as unmarked, should be captured with the same probability. Mortality rate should be the same. The marks should not be lost and should be easy to spot. And these organisms should be able to intersperse between each other at equally. So moving on, certain animals that can be surveyed using their natural marks. You do not need to place these external marks like dyeing them or placing certain chips or color banding them. These organisms, they can be you know measured, or the number of individuals can be measured, they can be identified based on their natural marks. So the basic one here is the tiger. So here is, you can see how they analyze the stripes on a tiger by performing this image analysis, and all of these tiger stripes, they are unique to the different individuals. Then there is this snow leopard, there is this clouded leopard, there is the common leopard, there's the marbled cat, striped hyena, spotted deer, the python, certain toads. In these organisms, if the stripes or the pattern on the body of the organism is unique between, say, different individuals, then you can use their natural marks to identify them and to carry out the census or the population survey for these individuals, for these organisms. Okay. So moving on, how do you measure the relative population density, or how do you indirectly measure the population density of different populations? So you can do this by placing these different traps, like the camera traps, or you can do these by fecal pellets. How many fecal pellets you find will be proportional to the number of individuals that are there in the population. Then you can carry out pug mark analysis, so you can also differentiate between the pug marks or where they are found, and based on that you can sort of estimate what is the population size. You can go out and you can listen to the vocalizations and the frequency of vocalizations. That will also tell you if what is the population size. There is pelt count. So when these organisms get hunted and their skin is sold out, so you can count how many pelts these hunters are selling or they have with them. That will give you an estimate of the population size. What is the catch per unit effort of trapping or fishing? For fishing, it is very easy. What is the catch per unit fishing effort? That will give you population size. Then what are the number of artifacts? For example, birds, they build nests. So what are the number of nests in that area? That will give you an estimate of the population size. You survey the field staff that is present in that particular region. So you go out and you ask these field assistants or the field officers that are present in that area, that how many of these particular species do you encounter every time you go out to the field. In that way you can get a rough estimate of the population size. Then if you divide that area into different quadrats, then what is the frequency with which the species is found in the quadrats? How many quadrats do you spot the species in? That will give you an estimate of the population size and the feeding capacity. How fast is the resource diminishing in that area? That will also give you an estimate of the population size. So this is the various ways of measuring indirectly what is the population size and the density. And what is the relative density? If you have two areas and you compare these different measures between those two areas, you will get to know if one of them has a higher population density as compared to the other one. So moving on, there are different ways in which you set up traps for insects. So I have shown here four different types of traps. So can you guess what are these traps which you carry out for insects to measure the population size and density of certain insect species? So what is this trap? What can you see here? in this image? Can you guys
The
guess?
first one is like a normal glue board, I think. Like the insect are attracted to that specific type of glue and they get stuck inside.
Yes, so you're correct. So the first one is a sticky trap in which you place some kind of an adhesive on that particular board, and then you hang it in the field. And this board is can be of different colors. Yellow is mostly an attractive color for different insects because they can be pollinators, or they can be getting attracted to the flowers of that color. And these insects, they will land on the trap, but they will not be able to get away from there. So in this way you can see how many individuals belonging to different species are getting trapped, and you can estimate what is the total population in that. Yeah, you can get a rough estimate of the population size in that area. What is this image showing? Which sort of a trap is this? Anybody? It was discussed in the lecture. So can you tell me what is this image showing? Okay, so if nobody knows the answer to this, so what we are seeing here is a pan trap. So you basically take different colored pans, and this is mostly used for pollinator insects. So the color of the pan signifies what is the color of the flowers to which these insects get attracted. So if some insects, they get attracted to yellow colored flowers, so you put out yellow pans in which you put water with some amount of either the nectar from those flowers, or these days people have started using different things like detergents and things like that. So these insects will land in the water trap, in the pan trap, and they will drown in the water. So they will not be able to come out. So they get trapped, and again, like a sticky trap, here also you can measure what are the different number of insects belonging to different species, and hence you can estimate the population size. What do we have here? You can like look at the image and guess what sort of a trap is this.
Like the insects are attracted to the light
Yes. So this is
and
called
they
a
get
light
trapped
trap.
in this.
At night you put out like a white sheet and you put out a halogen bulb, so the insects, they get attracted. A lot of moths, a lot of nocturnal insects, they will get attracted to the white sheet, they will come and sit there. And from here you can click images of the sheet, and then later on you can go and analyze the image, and you can get the number of individuals belonging to different species that were there on your light trap. Again, this is going to give you a rough estimate of the population size. And the last one, as it's written here, this is the pitfall trap. And how you create this is you take a plastic cup or a paper cup, and then you put it in the ground, and you put some water in that cup. And all the insects that are walking on the ground, like ants or beetles, they just stumble into the cup and they will not be able to come out. They will get trapped in the water. Another way of doing is you also take some stones and cover it with a lid, so that if it's raining, your trap does not get overflowed. So this will protect it from when it's raining or something like that. So can you tell me which of these is an invasive method of trapping insects, and which one is a non-invasive method? Out of these four different methods, which ones are invasive to the insects, are the invasive ways of getting to the population size, and which one is a non-invasive method? Anybody? Do you know what are invasive versus non-invasive methods? Anyone would like to take a guess from just the term invasive, non-invasive? Okay, so out of these three, the sticky trap, the pan trap, and the pitfall trap, these will be invasive methods. Why? Because these insects that once get stuck in these traps, they will not be able to escape and they will eventually die. So that is sort of an invasive method because you are harming the individuals of the species. And the light trap here will be a non invasive method because once you've taken the image, you can just turn off the light and all of these different nocturnal insects will just fly off. Another time, if you are actually capturing insects from the light trap, then that will also be an invasive method. But if you're just clicking an image of these insects and then you just let them go by switching off the light, then that is a non invasive method of getting to the population size and getting to know what is the different community of insects that are present in that area. So, moving on, talking about different ways of going out and sampling individuals in the field. So, one basic method that is performed is a line transit. So you basically decide a trail in the forest or in the grassland, and you decide that you're just going to look at the organisms that are present along this trail. So, say these are different, this is a deer population, and you find this trail, and you're just going to count the number of deer that you see from this trail. And so you go out and you see this first deer, and using your range finder you will find what is the distance of that deer from you. So how far could you see and spot the deer? Say this distance is d1. So this is the perpendicular distance from your line, from the transect that you have chosen. This is the perpendicular distance of the first deer that you saw. So you will say this distance is d1, and from here only you spot another two deers from here, and say the distance of that deer from the line or from you, from a point, is d2, and the angle between the transect and between the line along which you spotted the deer is theta, then the perpendicular distance of the deer from the line will be d2 sine theta, applying the normal right angle triangle rules. So if this angle is theta between the transect and the line of spotting, then the perpendicular distance of the deer from your transect will be d2 sine theta. So like that, you spot all the different deers and you get the perpendicular distances, and you can estimate what are the number of deers that are present in that particular area. So this horizontal distance you will know depending on how far you could spot a deer perpendicularly, how far in a perpendicular distance you could spot a deer. That would be say 50 meters on either side, and that will make the 100 meters. And you covered a transect that was 1 kilometer in length. So you would say that you have sampled this area of 100 meter by 1000 meters, and you have found this many number of deers in this area. Then you go out and you extrapolate this number to the total area of that region that you wanted to sample. Is that clear? This is how you carry out a line transit. So you now know that in this area of 1000 by 100 meters, there are say 8 deers. So in the entire area, there will be how many deers? You just multiply that with the entire area. Okay. So moving on, there is another method is called a point count. In a point count, instead of walking along a trail, you decide a point and you just stay at that particular point. So this individual stays at this one point and it is sampling the area around it. So this is the circular area that is around the individual. So if that individual stays in this point and is just turning around, it can sample all these different bird species. So all these red arrows show the birds that the individual was able to spot, and these are certain birds that the individual was not able to spot because either they were hidden in foliage or they are too far away for to see from the naked eye or from your binoculars. Again, from this point count, what you see is this is the area. So you know what is the radius of that bird from you. By using your range finder, you find out how distant is that bird, and you get the area of the circle in which you were sampling, and you can extrapolate the population size to the entire area. And you can combine these two methods by doing point counts along a transect. So you have a transect of 1 kilometer, and you're performing point counts at every 200 meters. So this is a way in which you can combine these two methods and carry out the population survey. Okay. So moving on, another method is a camera trap. So a camera trap. So this is a camera. You can see you have just tied it around to a trunk of a tree. This is camouflaged very well, and the animals will not be able to spot the camera. This is a highly non-invasive method. You are not um, influencing the animal populations at all. So certain images that you see in a camera trap are like this tiger, which has some prey in its mouth. Then there is this cat that you can see. There is a flying bat also that that is getting spotted in the camera trap, and there is this leopard. So from these images of camera traps, you can actually carry out the image analysis of the stripes or the rosettes of the leopard, and then you can estimate the population size in that area. So these are some ways in which you could sample the population size in a given area. You could do a line transect, you could do point sampling, or you could set up camera traps. So, some other sampling methods that are carried out to measure or to estimate the population sizes would be quadrat sampling that we discussed very initially in this session. There is distance sampling, which is also a type of transect sampling. So you decide the distance at which you're going to spot the organisms. That is the distance sampling. Audio strip sampling. So you move along a transect and you record the audio or the vocalizations that you can hear along that transect, and you're going to use these vocalizations in order to uh, estimate the population size. Then there is mark recapture, which we discussed. Pitfall funnel traps, we discussed them. There is this is the quadrat method. So these are the individuals or the crabs that you see within the quadrat, so you can count them. This individual, both of them are on the boundary, but you can count this because most of it is inside the quadrat, but you will let this go because this is mostly out of the quadrat. So there are certain rules or certain protocols that you will have to set up while you are using these different sampling methods. This is called a cover board survey, in which you use, say, boards made out of metal or made out of wood, and you basically place them and you dig out a little bit on underneath the board, and after different points of time you come and you lift up these boards and you will find different organisms beneath them. These can be insects, these can be, say, reptiles, amphibians that get attracted to the board, that are seeking shelter or cover. So this is called a cover board analysis. Then you're carrying out survey at the breeding sites. For example, you want to know the population of certain frogs or toads. So you can go to the breeding sites and you can count the number of tadpoles, or for insects you can count the number of larvae that are present in that area. For dragonflies, for mosquitoes, all of these, they can be used as survey at breeding sites. And there are quantitative analysis also that are carried out. So, population monitoring, why do we carry out population monitoring? In this, you get to know about the ecology and the behavior of the species, and you want to know the numerical abundance of the species. So, you're carrying out the population monitoring to know what is the ecology of the species, what is the behavior of the species, and what is the numerical abundance of that species in that particular area. And how do you carry out population monitoring? So you estimate the population size, and then you utilize the sex ratio and the juvenile to female ratio and the age pyramids. You use this, and then you can um, forecast, or you can predict what will be the population size. Is it going to increase? Is it going to decrease? Is it going to be stable across times? So you can carry out and you can make these different models in order to estimate what will be the population size over time. Then you can carry out this exercise at two different times as well, and then also you can know that, okay, at point A, when I went out to sample the population, there were X individuals. at point B, when you went out to sample the individuals, there were Y amount of individuals. So if X is greater than Y, the population is declining. If X is less than Y, the population is increasing. If X and Y are roughly the same, the population is stable in that area over time. Then, so in the lecture, we looked at two different types of growth curves. One is an exponential growth, one is a logistic growth. In the exponential growth, what you see is that the per capita growth rate, it stays the same. It does not change with the increasing population or over time. But in the logistic growth, the per capita growth rate, that is R, it gets smaller as the population size increases. So, what the exponential growth says is that in a particular area, the number of individuals that you find at, say, the generation 0 and is n naught, and the number of individuals that you find at generation 1, which is n1. So, n1 will be equal to r multiplied by n naught. So, every time in the next generation, the number of individuals are getting multiplied by r. So that is an exponential growth, and the graph will look something like this. It is J shaped, it is going to keep on increasing, and the formula followed here is dn by dt is equal to r max, that is the maximum growth rate multiplied by n. This is the continuous time formula, and the discrete time formula will be, if you integrate this particular formula, you will reach here. The population size at t is equal to the population size, the initial population size at generation 0 multiplied by e raised to the power rt. E is the exponent, the exponential power, the multiplied by the exponent rt. The logistic growth curve, on the other hand, is bound by the carrying capacity. It is more realistic because the resources are limited. The exponential growth assumes that the resources are unlimited in that area, but in reality, the resources will be limited. May it be space as a resource, may it be food as a resource, or may it be the amount of mates that you found. That will be limited, and therefore the populations mostly follow a logistic growth curve. The formula here is dn by dt is equal to r max multiplied by k minus n by k, which is entirely multiplied by n. So here k is the carrying capacity of the area of the region, the maximum number of individuals that that particular ecosystem can support. So here the R max, it stays stable, it stays constant, but here the R max or the growth rate, it reduces with the increasing amount of population. So when n gets higher, the R max multiplied by k minus n by k is going to reduce. So here the so here R stays equal to R max, because as you can see, both of them are highlighted in green. So R is equal to R max at all times. Here R is equal to R max multiplied by K minus N by K. So it reduces with the increasing population size. And there is no further increase in the population when the population is equal to the carrying capacity. When N is equal to K, this term will be 0, and hence there will be no change in the population size. So this is how the curve looks for a logistic growth. This phase in which it is growing slowly is called the lag phase. This phase in which the population is rapidly increasing is called the log phase. And when it becomes flat, equal to the carrying capacity, that is called the stationary phase. So this was all about exponential and logistic growth. Moving on to, this will be the last slide, the Lotka-Volterra equations. So these equations are used to estimate the prey predator populations in relation to each other. So here you can see that the formula is, here the predator population is not just affected by the number of predators, but it is also affected by the number of prey. So dp by dt, that is the change in the predator population, is equal to negative m multiplied by p, which is the predator population, multiplied by the death rate of the predators. So this is going to decrease the number of predators in the area, but if there are more, say more number of prey, then this is going to increase the number of predators in the area. So this is the birth rate of the predator or the prey. So here BH, which is the number of increasing prey, is going to increase the number of predators. So that is why this is a positive term in the equation. Similarly, for the prey equation, the birth rate of the prey is going to positively impact the prey population. Increasing births is going to increase the prey population, and increasing amount of predators is going to decrease the prey population. So these are the two populations, and when you plot them, you will see these two curves. So when the prey population is high, the predator population starts increasing, which leads to a consequent decrease in the prey population, and the decrease in the prey population is going to trigger a decrease in the predator population, and a decrease in the predator population is then in turn going to increase the prey population. So this is a cyclical relationship between the two populations of the predator and the prey. So this was the last slide of today, and if you guys have any doubts, please ask me. Please put it up in the chat box, or if you want me to discuss anything from the previous assignments that have been solved in the portal, so we can also discuss the assignments, any questions that you want. And if you guys have no further questions, you can leave the meeting. If you have any doubts, I am here and you can ask me. Either you can put it up in the chat box, or you can unmute yourself and go ahead and ask me if you have a doubt. What is per capita growth rate? Per capita growth rate is, so say the growth rate for the entire population is X, the per capita growth rate is what is the growth rate for every individual, or say per 100 individuals, like that. It is per unit amount of individuals. That is called the per capita growth rate. Is that okay? Is this clear? If the growth rate for the entire population is say X, the per capita growth rate will be what is the growth rate per individual of that population. Okay. Anything else? Any other doubts?
Ma'am, I have a doubt.
Yeah. Please go ahead.
Ma'am, like when you say that when the prey populations are increasing and subsequently the predator population will increase. So in I mean when we see in a sea happy there are lots of small fishes which are prey for big fishes like sharks and whales. But we don't see exponential growth of sharks or whales. We only see a finite amount of sharks or whales in our
Yeah, they will not
oceans.
increase exponentially, because once they consume a certain amount of prey population, once they consume a certain amount of small fishes, the population size of the small fishes is going to decrease, and eventually there will not be enough prey for the predator, for the big sharks, and that is going to lead to a decrease in the shark population, and this is going to allow the prey populations to flourish again. So it is a cyclical relationship. If there is more small fish, the shark populations will increase, but the increase in sharks is going to lead to a decrease in the small fish, which is consequently going to limit the number of large fishes or the sharks. So you will never see the sharks go like exponentially increase, because that would, they are controlling their own population, because they are consuming the prey in high amounts, which is going to reduce the prey that is available for them in the future.
But if that's the case, then we humans are also in that cycle. No, ma'am. Like we are also a factor for the reduction of the prey percentage in the sea. Like we are fishing out a lot of small fishes, which will, affect sharks in a way, right?
Yes, that is what
So,
is happening. So humans are going out, they are fishing, they are capturing a lot of these small fish, and this is in turn reducing the amount of prey that is available for the large fishes out there. So it does affect the large fish, and their amounts get reduced because of us capturing all these small fish that is their food.
oh, okay. So like it balances out automatically. Okay.
Yes.
Yes, ma'am. Thank you.
Thank you. Any other doubts?
No doubts, ma'am. Thank you, ma'am.
Thank you. Okay, so if there are no further doubts, I will end this session. Thank you.